subscribers, a very good evening to all of you. I mean, good afternoon, all of you. Yes, once again, we are here with the practice session of DNA. Yes, children, in the previous few sessions, we have started our molecular basis of inheritance and we have discussed a lot of things about the DNA, the structure, the genetic material, the research, the experiments performed to find out that whether the DNA was a genetic material or not. So, I thought why not solve questions related to it. Yes, since we have studied so much about it, let's solve few questions related to it so that you understand that whether you have understood the concept clearly or not. Yes, again we are here. Let me introduce you to myself. I am Anam Khan. I'll be your botany expert for the session 2023 and 2024 and we're going to do all the amazing and interesting topics related to biology. Yes, of course, like we have already completed one chapter which is the sexual reproduction in flowering plants. We are on the next chapter which is the molecular basis of inheritance. Yes, it's a very interesting chapter. It's a very conceptual chapter also. Yes, today there might be a practice session of the DNA, but tomorrow at the same timing we're going to discuss about the RNA world. Yes, we're going to introduce you to the types of RNA existing, what is RNA, what are the features of RNA in detail, just like we have studied about the DNA. So, without wasting any time, let's start today's session, yes, which is the DNA practice session. Talking about the DNA, we have, of course, you know this chapter is going on in our class, which is the molecular basis of inheritance, and the topics to be covered in this chapter is these: the research for the genetic material, the DNA structure and packaging. Yes, today is about the DNA structure and packaging practice session. We are going to also discuss about the RNA world, replication, transcription, genetic code and gene mutation, translation, regulation of gene expression, human genome project and DNA fingerprinting. Yes, we are going to discuss all of these in the coming session. So, yes, without wasting any time, let's see what we have in today's session. Let's see what are the questions we have to solve. The very first question is here. Yes, the pneumococcus experiment proved that, yes, remember we used the pneumonia, Diplococcus pneumonia, yes, experiment. What does that prove? Yes, it's a very simple question. We have discussed about this in detail in the class. What does the pneumococcus proves? The experiment of Diplococcus pneumonia, what does it prove? That DNA is a genetic material, DNA undergo binary fission, DNA bacteria do not reproduce sexually, or all time RNA production of DNA and protein. What does it prove? It's a very simple question, I would say. Nothing could be simpler than this. Yes. What could be the answer? Yes, take your time, read the question and then answer carefully. Yes, DNA, what does the pneumococcus experiment proves? Yes, the pneumococcus experiment proves that DNA is a genetic material. Yes, it proved that the transforming principle was the DNA, something which actually transformed the non-virulent to the virulent nature. Yes, we have discussed about this in detail. It was given by Griffith, transforming principle, which was further explained by MacLeod, McCarthy and Avery. Yes, next we have another question. The smallest unit of genetic material which when mutated produce a phenotypic effect. You know what is genotype? What is phenotype? Genotype is description of the genes. Phenotype is what is basically going to express, how it is going to express the external appearance. Yes, that is the phenotypic. So, children, if the smallest unit of genetic material when mutated produces a phenotypic effect, what would be that? Yes, cistron, muton, recon or nucleic acid. Put your little bit brain into it and you will be able to answer. Yes, everyone. The smallest unit of genetic material which was mutated produces a phenotypic effect. Yes, the smallest unit is the muton because the mutation is going to happen in the muton only. Next question. Who coined the term gene? Yes, who coined the term gene? The gene is the, what is the gene? Weldwer, Mendel, McClintock and Bateson. Simple. Who coined the term gene? Genes which are present on the chromosomes. The chromosomes are the carriers of the genes. Yes. Who coined the term gene? The correct answer is the Walder. Walder coined the term gene. Very simple. Next we have the strongest evidence that proves that DNA is a genetic material coming from. Yes, what is the strongest evidence that the DNA is a genetic material and it is coming from? Quickly read the question carefully. The answer line is in the question only. Very simple. Strongest evidence that the DNA is a genetic material. Quickly. Yes, the fact that the chromosomes are made up of DNA, the findings of DNA is not present in the cytoplasm, the knowledge of DNA is present in the nucleus, studied of transformation of bacterial cells. Yes, which proved that DNA is a genetic material. Yes, the correct answer is the studies on transformation of bacterial cells. Yes, the transformation in the bacterial cells shows that the DNA is a genetic material. Simple. Next we have the Griffith experiment was based on simple. This is absolutely simple. The Griffith experiment was based on what? Yes, the Griffith experiment was based on what? The Griffith experiment was based on was actually which particular organisms proved that the transforming principle, and he performed with yes, the correct answer is the Diplococcus pneumoniae. Yes, the Diplococcus pneumoniae was a Griffith experiment performed by the Griffith on this to see the transformation, that it was a virulent or not virulent. Yes, next question. The principal genetic material of a living being are, what is the principal genetic material of living being are? RNA, DNA, both A and B, none of these. Yes. What is the principal genetic material of living being? Principal genetic material of living being. Yes, what is the principal genetic material of living beings are? RNA, DNA, both A and B, none of the above. The correct answer is DNA. Yes, DNA is the genetic material of a living being. RNA is not. Next question. Hershey and Chase experiment with bacteriophages shows that, what does the Hershey and Chase experiment with the bacteriophages shows that? We have already discussed about how, yes, marking the phosphorus radioactive and of course the sulfur radioactive. We have seen that how the transformation is happening through the bacteriophages, which are basically infecting the bacteria. Yes, if you have missed it, how basically this experiment has taken place, there's already a class scheduled. Already we have taken this class. It's already present in your past Embibe classes. Whenever you have time, you can go through it. Yes. Hershey and Chase experiment with bacteriophages shows that, very simple, what does it shows? Hershey and Chase experiment shows that DNA is the genetic material. Protein was typically discarded. Yes, and of course DNA was the genetic material. Next question. The final proof of DNA as a genetic material came from the experiment of, final proof. Who was very much successful to prove, yes, without any doubt, DNA was the genetic material. Just without any doubt, it was confirmed that DNA was the genetic material. Yes. Who did that? Hershey and Chase, Avery, McLeod, McCarthy, Harbovind, Khurana and Griffith. Who did that? The final proof for DNA as a genetic material came from the experiment of Hershey and Chase, Avery, McLeod, McCarthy, Hargobind, Krava and Griffith. Who did that? Simple. Hershey and Chase experiment finally proved that the genetic material was the DNA using the bacteriophages. Next. Transformation was discovered by, who discovered the transformation, that this transformation is basically happening, converting the virulent, non-virulent into the virulent. Yes. How this transformation is happening, it was discovered by whom? Yes. Who discovered the transformation? Who discovered the transformation? Michelson and Stoll, Hershey and Chase, Griffith, Watson and Crick. Yes, the correct answer is the Griffith. The Griffith was discovering the transformation experiment. Very simple. Next, 1944, Avliard McCarthy isolated a substance from the heat-killed virulent form of bacteria, added to the non-virulent bacteria, which changed the non-virulent bacteria to virulent from the substances can be destroyed. Yes. Which change the non-virulent nature into virulent from the substances can be destroyed through. Yes, the changing mechanism was disrupted by which enzyme? Which enzyme was able to disrupt that transformation? Yes, which enzyme was able to disrupt the transformation? Yes. The answer is the DNases were there to disrupt the transformation of non-virulent to virulent, and it was discovered Appleyard, McCarthy and Avery, and they got the Nobel Prize for that also. Next question. In the bacteriophage experiment, Hershey and Chase demonstrated that DNA is the genetic material in, yes, in bacteriophages experiment, Hershey and Chase demonstrated that DNA is the genetic material in TMV, Escherichia coli, T2 bacteriophage, Diplococcus pneumonia. Yes. So children, the Embibe app gives you an opportunity so that you can directly answer questions through your laptop, your mobile. So please, which is not present on the YouTube, so I request you to please join us on the Embibe app so that you can answer directly and you can see all the particular session have been scheduled. Yes, in the sequence, you can register yourself for the upcoming classes so that you never miss an update. And before coming to the session, you can always go for the pre and the post class activity so that you know whether what is going to be taught in the class, and after the class gets over, you know what is being basically discussed so that you can solve questions related to it. Yes, in the bacteriophage experiment, Hershey and Chase demonstrated that DNA is the genetic material and he uses T2 bacteriophage. This experiment is absolutely important from your examination point of view, and if you ever wanted to give the NEET examination, if you wanted to basically sit for the medical examination, this whole chapter is absolutely should be on the tips. Yes. Next question. Experiment of Griffith on mice explained the phenomenon of transduction, replication, conjugation and transformation. Experiment of Griffith on mice explained the phenomenon of what? Experiment of Griffith on mice explained the phenomenon of transformation. Yes, the Diplococcus pneumonia which Griffith performed on mice explained the transformation principle. Next question. A nucleoside differs from a nucleotide by not having, this was a very important thing when I was explaining you nucleoside and nucleotide, I was telling you that this is a difference. What does a nucleoside differs from a nucleotide? What is the difference between both of them? There is a sugar, there is a glucose, nitrogenous base or phosphate group. Very simple. Yes, quickly see, quickly answer this. Yes, the correct answer would be the difference between both of them is there is a phosphate group difference. Nucleoside differs from a nucleotide by not having, there is a no phosphate group. Next we have another question. The anti-parallel nature of DNA refers to as, yes, The anti-parallel nature of DNA refers to as, how we are basically saying that these are having the anti-parallel nature. It is charged phosphate group, the formation of hydrogen bonds between bases and opposite strands, the opposite directions of the two strands, the pairing of bases on one strand and with bases on the other hand. Yes, what could be the answer? Anti-parallel nature of DNA refers to its charged phosphate group, formation of hydrogen bond between bases and opposite strands, the opposite direction of two strands, the pairing of bases on one strand with the bases of another hand. The answer is the opposite direction of 5 to 3 prime, 3 to 5 prime. That nature refers to as anti-parallel. Next question, double hydrogen bonds occur between DNA of uracil and thymine, thymine and cytosine, adenine and guanine. Double bond is between, remember ATGC pairing, the double bond will be present in adenine and thymine. The cytosine, adenine, ATGC, the guanosine and cytosine will have 3 pair, but 2 pair will be present in the adenine and thymine. Next question, new strands of DNA are formed in the direction, the new strands of DNA will formed in the direction, the new strands of DNA are formed in the direction the new strands of DNA are formed in the direction of 5 to 3, 4 to 3, 3 to 3, 2 or 3. Yes, it's always into 5 to 3 direction. Simple. Next, the DNA nucleotides are attracted by, how these two nucleotides are basically polynucleotide chain has been formed. How these two nucleotides are attracted towards each other? Through hydrogen bond, covalent bond, van der Waals bond or electrovalent bond. Which bond is responsible for the attraction? Yes. Which bond is necessary for the attraction? Hydrogen bond, covalent bond, van der Waals bond, electrovalent bond. The answer is the hydrogen bond is responsible for the attached nucleotide. Yes. Next question. In DNA strands, the nucleotides are linked together by, how these nucleotides are linked together? How these nucleotides are linked together in a DNA? Yes. How these nucleotides are linked together? How these nucleotides are linked together? Glycosidic bond, phosphodiester bond, peptide bond or hydrogen bond? Yes, they are basically linked by the phosphodiester bond. Yes, the DNA strands are linked by the phosphodiester bond. Next question. With whom did Watson and Crick share the Nobel Prize for DNA structure? Yes, with whom did the Watson and Crick share the Nobel Prize for DNA structure? They actually shared the Nobel Prize for DNA structure with the Wilkins. Yes, the Watson and Crick shared their Nobel Prize with the Wilkins. Next question. The fact that a purine base always pair with the hydrogen bond of a pyrimidine base in the DNA helix leads to, yes, A purine will always pair with the pyrimidine. Yes, ATGC. This is how the bonding was done. It was because to have a uniform width throughout the DNA. The reason that the purine will form a bond with the pyrimidine so that they can basically uniform width throughout the DNA. Next, in a DNA strand, the nucleotides are linked together by how these nucleotides are linked together through glycosidic bond, phosphodiester bond, peptide bond or hydrogen bond. Glycosidic bond, phosphodiester bond, peptide bond, hydrogen bond. How these two nucleotides are linked together? Very simple, just now we have studied. It is linked through the phosphodiester bond. DNA strand nucleotides are linked together. Yes, through phosphodiester bond. Next question. DNA as an acidic substance present in the nucleus was first identified by Dash in 1869 and he named it what? Very simple, we have also discussed this. The DNA was an acidic substance present in the nucleus was first identified by yes, Mixture, Nuclearin, Watson and Crick DNA, Chargraph, Nuclearin, Wilkins and Franklin. Very simple, it was Mixture. Friedrich Mixture actually found out that is in the DNA as an acidic substance and he named it as nuclearin. Next, the nitrogen base of DNA is, what are the nitrogen bases? Simple, ATGC, purines and pyrimidines. Next, the substance that act as a connecting link between the two generations. What is a connecting link between the two generations? Yes, what is the connecting link between the two generations? It has to be DNA, deoxyribose nucleic acid. Next, Watson and Crick and Wilkins were awarded the Nobel Prize in Medicine in the year, in which year they were awarded the Nobel Prize? Yes, 1861, 1862, very simple, in 1962 they were awarded the Nobel Prize. Next, in eukaryotes, the genetic material is, what kind of a genetic material present in the eukaryote? It is circular DNA with histones. Yes, there is a histone protein. Next, in prokaryotes, the genetic material is circular DNA without histones. Very simple. Next, so children, this is all about today's session where we have solved questions related to DNA. What is this? This is your Embibe app. Yes, you can log in through your email ID and your phone number. What you have to do, just click on the side button, select your exam which is your board, which is could be any board you belong to, CBSE, and then you press on the 12th CBSE, select your respective language and press done. You will land on this page and as you click on the home page button, go for the Embibe live classes, you will land on this page and as you can see children, I have already, the next session will be about the RNA world. We are going to discuss about the RNA in detail. So, do not forget to join us for that session. Yes, all the upcoming session has been scheduled. Yes, as you can see, these are all the sessions which have been scheduled for you. So, yes children, you can learn, you can practice, you can test through this particular app. And there is another app which is the Embibe Lens app. What you have to do, you just have to scan the text, highlight all the keywords and will see everything in the 3D. We'll continue discussing about the molecular basis of inheritance. We have a lot to discuss about the packaging of the DNA, the replication of the DNA, the RNAs and everything. Until then, we meet again. Until then, take care, keep imbibing. Bye-bye.
Keep imbibing. We believe in you.